everyone. Welcome to a Moment of Science, Please. My name is Ellis, and I will be your host for today's episode. A Moment of Science, Please is a student-run podcast group based at the University of Toronto. Our goal is to highlight some of the fascinating scientific research we have going on here. We have joining us today Dr. Jennifer Mitchell, a professor in the Department of Cell and Systems Biology. Welcome, Dr. Mitchell.
Thank you for having me today.
Could you please start us off by introducing yourself and telling our audience a little bit more about your background and research?
Sure, yeah, no problem. In terms of my background, I did my undergraduate degree at the University of Waterloo, which is about an hour away from here. I was in a co-op program there, so I was able to take placements in different types of jobs over the course of my degree, which really helped me to figure out what I wanted to do next. And my favorite placements were in two different university research labs, which was then why I decided to pursue a PhD. I did my PhD here at the University of Toronto at a research lab in Mount Sinai Hospital, where we were studying pregnancy and labor. And after that, I went to Cambridge, England to do postdoctoral work for 6 years. And then I came back here to start as an assistant professor at the University of Toronto.
How did you choose your area of research specifically within the field of genomics and developmental biology?
Yeah, so I think it's really challenging initially because you're learning so much as an undergrad, right? There's just so many things that you learn. You might be studying across a really broad array of disciplines. But when I was in my final year, or maybe even a little bit before that, I just started really reflecting on what I found the most interesting and, started to just really think about where my real passions and interest might lie. And I realized that I really liked developmental biology, but I was just really fascinated by how changes in gene expression could direct that complex process. And so my whole career has really focused on investigating the mechanisms of how genes are regulated. So how genes are turned on and off in specific contexts. And that passion then kind of led me more into genomics as, the field kind of grew and changed to trying to better understand how whole complex genomes function and kind of taking a broader view. But I really started with a focus on trying to understand how one gene was regulated and then, sort of broadening out from there.
Was it hard to find where you wanted to do your research specifically, or was it kind of a smooth path?
Yeah, I think it is challenging because there's lots of points where, you make a decision that sends you down a certain path. But just because you do your PhD in one area doesn't mean that you're stuck in that area. So, I did my PhD in an area of women's health. We studied pregnancy and labor, but I was working on a question of how a particular gene was regulated during pregnancy. And so when I went to change and do postdoc work, I sort of kept with the gene expression and gene regulation aspect of my career. And I left to the side for a while that sort of reproductive biology and women's health area of research, which I've actually come back to more recently. So things can kind of, come full circle as well. But sometimes we do think that, we can't change our focus over time, but you really can. You're never stuck in one area. That's one great thing about, research and academics in general is you're always kind of pushing into new areas. So you're always trying to do something new, which can be really challenging, but it's also just really exciting.
Mm-hmm. That's a great lesson to learn. You don't always have to keep within your comfort zone when you're testing out the waters.
Definitely not. Definitely not in a comfort zone usually.
For sure. So one of your recent papers is titled A Flexible Repertoire of Transcription Factor Binding Sites and a Diversity Threshold Determines Enhancer Activity in Embryonic Stem Cells. Could you give us a brief summary of what this research is about and how the methods and approaches are used within the study?
Sure, yeah. So in this paper, we were studying how embryonic stem cells, which are a specific type of stem cell that can differentiate into any cell type that makes up the mature body plan. So they're a very special type of stem cell. We were studying how a specific gene regulatory element called an enhancer functions. So we used computational biology approaches, and specifically we were trying to investigate the sequences in the genome that encode those specific type of regulatory elements. So enhancers, like the name sounds like, enhance or they increase the amount of any specific gene that's being transcribed and translated into protein, which is often the functional unit of a gene. So for example, we know that in our red blood cells, specific enhancers cause our cells to produce a lot of globin, and that then allows our red blood cells to carry oxygen throughout our bloodstream. But actually, we still don't have a really good understanding of how each bit of the sequence within those enhancers functions. So we were trying to better understand these really important gene regulatory elements in our genome at a sequence level. So we use computational approaches to try to better understand how the sequence encodes function in these enhancers. And using these computational approaches, we actually developed such a good understanding of these sequences that we were able to build functional synthetic enhancers from small building blocks of DNA. So we were, sort of trying to test our understanding by building them from first principles. And we were pretty excited when it actually seemed to work. So we felt that really demonstrated how we had learned more about these important regulatory elements.
Interesting. Now, looking at this field as a whole, what would you say are some common misconceptions of research within this field?
So I think, if we're talking about the general public, I think, there's a common misconception that we've already solved a lot of these complex problems when really actually we've only scratched the surface. So, we sequenced the human genome almost 20 years ago, but we still don't know what a lot of that sequence is used for in our genome, right? There was new genome editing technology that emerged in 2012, which is the CRISPR-Cas system that lots of people have heard of. And that now allows us to change genomes in ways that are more efficient than ever. And there's a lot of excitement for using this as a treatment for genetic diseases. Also, lots of concern about what some of the unexpected side effects might be. But what I think most people don't appreciate is that for many situations, we don't actually even know what we'd need to change to cure someone of a specific genetic disease, right? There's only really a few examples where we really have a lot of details. And so, we don't really know enough about what most of the sequences in our genomes are doing. And so we're still missing such a lot of fundamental knowledge. And, I think why people are often not aware of that is that, across a lot of fields, is that often new advances are reported by the media in pretty simple terms, right? And a lot of the real meaning behind the advance is left out. So there usually isn't real discussion about what we still don't know when a new advance is highlighted. And so over time, I think that leads to a false impression of where we actually are in most of these areas. And I was just thinking of, the example of something like a cancer treatment, right? So you might hear in the news, oh, there's this really new, exciting, transformative treatment. And there's a lot of excitement about it. But then if you go and actually look at the underlying research, that treatment may have only been done in mice so far and might not even be approved in humans yet. And, it could be another 5 to 10 years before we know if it even works in humans, right? And that's kind of a common theme. Then next week you might hear about some other new cancer treatment, right? And so I think over time, that builds the false impression in people that we should have solved this problem by now, right? So people start thinking, well, why is cancer still an issue if I'm always hearing about new treatments, right? When they don't really realize that those treatments aren't even often approved in humans yet, right? So I think sometimes there's an oversimplification of these complex problems when they're reported on, and over time that kind of leads to this false impression of where we are in many fields, especially in treating human diseases, right?
Out of curiosity, then, how would you say does ethical concerns and regulations come into play with this field of research?
Yeah, I mean, there's been a lot of talk. There's lots of different ethical issues. So, there's issues with thinking about how, for example, countries that don't have centralized healthcare deal with things they consider pre-existing conditions when they're genetic diseases. Um, there's issues concerning how things like 23andMe or other online genetic sites that will provide you with some genetic information are actually using that information, and how they're counseling or not counseling people to think about it. So yeah, I think there can be a lot of issues when we think about how we deal with any information that we might learn about ourselves based on our genetic makeup. Because again, not only do we not really fully understand how the sequence of our genome causes, different diseases or different phenotypes or things about ourselves, but we also don't fully understand the interplay between the environment, right?
For sure. And what role would you say that misinformation would play in this picture?
I guess again, it comes back to thinking that things are simple, right?
Hmm.
Yeah, I mean, there's different types of misinformation, right? There's just sort of misinterpretations or like oversimplifications that are not necessarily meant to be malicious. But then, we have over the past several years encountered a lot of problems with more deliberate misleading of people using things like social media and to influence people. And I think that's a problem across a lot of disciplines and not really restricted to biology, right? Um,
Yes.
but I think overall, it kind of underscores really the importance of education, right? The more educated our general public can be about science, about, just about anything, about thinking about complex problems, then I think the better, we are able to deal with things like that, right?
Yes.
It's also important to know where to go for trusted information. So often, when I'm teaching courses and I'm in addition to giving people the information that I think is important, I'm also trying to point them to the places to go to get more information where it's a trusted source, right? So I teach a course in
Hmm.
stem cell biology, and one of the things I do is direct people to websites that have information about stem cell research and stem cells in the clinic, and what types of questions to ask if you're considering using or being part of a stem cell clinical approach. And because I want people to be able, who've taken my course, to be able to educate their friends and family about where to go to find the right information, right? So, I think it's important to know where to go to find those trusted pieces of information.
So then at the current stage, what would you foresee becoming a popular direction of research in your field? And why would you say so?
I think it's really hard to predict the future. And so, yeah, and so my thoughts on this are not really kind of future prediction, but just to say that I think advanced computational approaches like machine learning and using computers to solve really complex problems is only going to continue to grow, especially in the field of genomics, where we just have so much information. Genomes are huge, right? Even small genomes are huge. And so you really need those computational approaches to try to understand, try to draw meaning out of the sequence, right? But these, machine learning approaches are bleeding into many fields at the moment, and, you know, helping us to make sense of larger and larger amounts of data that we need to try to understand.
Got it. Okay, so do you have any tips or advice for students who are looking to gain research experience either during the school year or during the summer?
Yeah, so I would say, kind of similar to what I mentioned about my journey, I would say just really take some time to think about what area really excites you. And so, think about what you've learned throughout your undergraduate and what you found the most interesting and exciting. And then look for people working in that area of research. And, University of Toronto is a really big university, and that can have its challenges. But because we're such a huge research center, there's just so much going on here. So regardless of what your interest is, you can probably find somebody working in that area of interest. But really, I think it's best to start with what specifically interests you, right? And then once you figured out what interests you, I'd say just try to get involved in the research. Try to be a sponge, watch, learn, read the literature, and offer to help with anything that's related to the experiments. And that's usually how everybody gets started, right? And then, it can seem initially like you're not doing a lot, but even really small tasks can build up into big contributions. So, just and keep persevering, keep learning new things.
Okay, so one last question. We ask all of our guests this question. Do you have a favorite scientist? And if so, why?
My favorites probably fluctuate over time. I'm starting with that because I'm going to mention two people that more recently became well known. And so I think at the moment I'd have to mention as two really incredible women, Jennifer Doudna and Emmanuelle Charpentier, who shared the 2020 Nobel Prize for their discovery of the game-changing gene editing technique, which is the CRISPR-Cas system. And I just think they're both so amazing for so many reasons, but one of which is that, they were working in this field that many people thought was obsolete. So they were working in a bacterial genetics field. And, many people in this field were struggling to get funding because people had a hard time seeing the usefulness of continuing to study bacterial genomes when we'd sequence the human genome. And so not only did these, two women and their research labs, because of course we all work in teams, but not only did they solve how most of the life on our planet keeps its genome safe from invading viruses, because the CRISPR-Cas system is sort of like an adaptive immune system, but they also discovered this amazing genome editing technique that's revolutionized the way most of us are doing our work, and it has huge potential in the clinic. And so, their work has been so transformative, but I think they're also just such great role models for young women. And the prize was actually awarded fairly quickly for a Nobel, right? So from discovery to award was only about 8 years. And I think that really underscores how transformative this finding was for biological science. And it was so amazing to see them share this prize, for so many reasons, but also because, I'm gonna get tearing up here. So many women in the past have been overlooked for their contributions to science. And so to have that 2020 prize go to these two amazing women was just so great, such a great moment, right? Um, and, now, ooh, very choked up yeah. So I also see them, they're just both such, Dowden and Charpentier, both such amazing, great public speakers. And so they're really great role models for young women. And they're also both advocating for like a very considered and careful approach as CRISPR moves towards clinical application. So I guess I just can't really admire them more. I think they're just so amazing. And it was so great to see them get that prize. So yeah, though that's who I'm sort of thinking of right now as my favorite scientists at the moment.
So with that, this concludes our podcast. Thank you everyone for listening. If you found this podcast interesting, please take a look at our previous episodes. Keep an eye out for new episodes, and don't forget to subscribe to us on YouTube and follow us on social media.